So in this video, let's see what are the consensus sequences. See, consensus sequences are frequently and consistently occurring sequences in the DNA. In this video, let's see what is the sequence of INR consensus sequence. See, INR consensus sequence is present in the case of human beings. It can be written as YYA and WYY. As we know, all the consensus sequences are written with respect to the coding strand from 5' to 3' direction. So, suppose this is a template strand for the transcription and this is the coding strand. So, this one is the non-coding strand. This is the coding strand. TSS is our transcription start site. INR consensus sequence is YYA and WYY. This is in the coding strand. See, we have shown it in the other strand than the template strand. So, this strand is the coding strand. This is our INR sequence from 5' to 3' direction. It is YYA and WYY. Y is pyrimidine. This is also pyrimidine Y. A is the adenine present at the +1 position. See, this position is corresponding to this +1 position. So, when adenine is present here at the +1 position, the corresponding nitrogenous base here will be thymine. This is actual transcription start site or +1 site. Because adenine is present here in the coding strand, actually here will be thymine. After that, in the sequence is N. N can be any nucleotide. W is any weak nucleotide, that is nucleotides that contain adenine or thymine as their nitrogenous base. Why such nucleotides are called weak nucleotides? Because A and T, these two nitrogenous bases are attached to each other with the help of 2 hydrogen bonds, and G and C are attached to each other with the help of 3 hydrogen bonds. So, A and T, all those nucleotides which contain adenine or thymine as their nitrogenous base can be called as the weak nucleotides. So, here will be either A or T. Y is the pyrimidines. So, what is the sequence of nucleotides in INR consensus sequence? First is the pyrimidine, then adenine, then there can be any nucleotide, then here will be either A or T, here can be any pyrimidine. Now, as this sequence is written with respect to the coding strand, actually in the non-coding or the template strand, the sequence will be like this. If this is pyrimidine, here can be any R. R is the any protein. Here is the pyrimidine. So, in the non-coding or the template strand, here it will be any purine. Here will be purine. Other one is also purine. At +1 position, here will be thymine. Because in the coding strand, here is adenine. So, in the template strand, here will be thymine. Then there can be any nucleotide. After that, there will be any weak nucleotide. Because if here is A, then here there will be thymine. If the thymine is present here, then in the template strand, here will be adenine. Similarly, these are 2 Ys or the pyrimidines. So, in the template strand, here will be 2 purines. So, let's see what is the sequence actually present in the template strand. It will be purine at -2, purine at -1, thymine at +1, any nucleotide at +2, any weak nucleotide at +3, purine at +4 and also purine at +5. So this is the actual sequence present in the INR in template stand, but while we write it with respect to the coding stand from 5 prime to 3 prime direction. So it will be YYA and WYY. So this is the sequence of nucleotides in the INR consensus sequence.